to this point before we go along to the next point. Okay. And that kind of rationalization fits. Because this is immunology, we follow this progression, these different stages in, of course, looking at surface markers, okay? And because we're talking about B cells that rearrange genes, another assay we use is the status of genes. Are they rearranged or not? Are heavy chain genes rearranged or light chain genes rearranged? And we saw this figure earlier, looking at a number of criteria in staging cells at these different points. And RAG, remember, is the recombination activating genes. TDT is terminal transferase, the enzyme that attaches nucleotides in a non-templated way. The only enzyme that does that, I believe. IL-7 receptor. B220, interesting story about B220. So B220 is often used as a B cell marker when one is doing gross sorting of a mixture of cells and looking at T cell versus B cells such. B220 is often used as a way of selecting for or selecting against B cells. Interesting thing is that it's actually an isoform of CD45, which is common on, I think, all white blood cells. And another marker that's used for CD19, widely used in flow studies, again, when you're looking at all B cells, or you want to remove all B cells, CD19 is another important one used for that. And we see here where that begins to be expressed. And we see B220 from the beginning. So pre-pro, remember that nomenclature, and again, I'll state, I guess as I did the other day, that we'll use the nomenclature of Basel and Melcher's. Basel is the city, the institution, and Melcher's is a scientist. I realize if you don't know either in that way. If you're not familiar with Switzerland, I know that. And then there's also the Hardy system, which is described in your book. So again, B cell lineage marker, B220 in the pre-pros. They receive signals that are through the IL-7 receptor that are important, and just thinking logically, well, at the earliest stage, the genes are still in the germline configuration, as makes sense. So pro-B for progenitor, different transcription factor expressed, and now the early D to J in the heavy chain locus has occurred. And by, they call the late pro-B, there's some V to DJ. And now CD19, the marker is present. By the level of pre-B, now V to DJ has occurred for everyone. And so again, this makes sense, the order that we've already gone over and staging these cells like this. And now, because the VDJ has occurred, now there is the membrane expression of the pre-BCR, so that the heavy chain has rearranged, can be expressed, and with the surrogate light chain, there's a pre-BCR on the surface. These large BCRs, large pre-Bs can now undergo a bunch of divisions and gives a chance for different heavy chains, the same heavy chain to be paired with different light chains in different cells.
question.
Yeah.
So when we're studying this material, would you recommend attempting to memorize all those status of
Certainly
gene
not
changes?
all of these.
Okay, 'cause
Maybe
my
the
brain's
status of
swimming.
immunoglobulin genes.
Okay.
So these large B cells are proliferating, they're large, and those that don't rearrange well, they go through apoptosis. So checkpoint here. So now there's a new pool of these cells, all with the same heavy, and they can rearrange different light chains. They now stop proliferating and become these small pre-B cells. And there's many chances to have a successful light chain rearrangement. In mouse, first you can try one kappa, and then if that doesn't work, the other kappa, and then lambda. In humans, the kappa and lambda have equal chances from the beginning. Once a light chain is made, now there's IgM on the surface, and that stops light chain rearrangement. So this is now called an immature B cell. Again, notice if light chain doesn't work, then at least can lead to apoptosis, but also editing is possible, especially at this locus of the light chain. So in other words, a further rearrangement to put a new V and J together. Slide on pre-B cell receptors with the SLC stands for surrogate light chain. So many chances to get this right between in mouse the kappa and lambda loci. One chromosome has a chance to arrange correctly in each of those. Each first and second in kappa, first and second in lambda, and then editing can actually occur beyond that. So many chances to get it right and not undergo apoptosis. So let's see questions here. So now we're up to the immature B cells as shown in this figure, where we've rearranged both the heavy chain locus and the light chain locus, and now IgM is expressed on the surface. So I guess we can go through all these. Pre-Pro B first, and going into to early Pro B, late Pro B, large pre-B, then small pre-B, then immature B. And this is the kind of thing one should know. So pre-Pro B is the configuration is germline, immature B, everything is rearranged, and so in the early Pro B, DJ rearrangement, and then large pre-B, the V to DJ and the heavy chain. Okay, so now the same issues, some of these, our story has been parallel to T cell development with the rearrangement, following markers on the surface, but we also have this big picture. What do we need to do? We need to have tolerance and have a way of removing all, or at least some, of the cells that are going to be self-reactive because of this great diversity that we have achieved. So these guys are very sensitive to tolerance induction. They bear these markers on the surface. They're tested against self-antigen. All these things, there's experimental evidence for all these ways of limiting, of having tolerance and limiting autoreactivity. Clonal deletion, receptor editing by a reactivation, a recombination, or energy. Energy used here and in general being not being able to respond. Having the machinery to respond, having the appropriate B cell receptor or a T cell receptor, but not being able to respond for whatever reason. So we call these kinds of actions clonal deletion, receptor editing, central tolerance, similar to what we talk about deletion of thymocytes due to recognizing self-antigens. We call that central tolerance. So how are we going to address this? So let's think of back to what was used with T cells, and same idea, many of the same framework for thinking about this applies. So we have the issue of, with the immune system, great diverse repertoire, how do we follow each individual B cell? And the answer, as you know by now, is using transgenes, looking at transgenic B cell cells transgenic for the B cell receptor, in which all or nearly all will express the same B cell receptor, and thus we'll be able to follow that one, follow those B cells. So the approach that was taken, and the main person for this, as mentioned in the book, is Namazi. That's how to say the name. And so he did these experiments. He's at, or was at, back when I was doing immunology, the National Jewish Medical Center in Denver, which little story about that's not like a real, it's not Harvard or Stanford, a really famous place, but for immunology, it's a really, really top place. And I think a couple times in the book already, the labs of Pippa Marek and John Kapler have been mentioned, and they're there as well. So that's a real great center for immunology. So his approach was, again, transgenic mice, all expressing the same B cell receptor, and also made use of an antigen that could be, and I should say what the antibodies were recognizing a class 1 molecule. So that way, see the analogous situation with the T cell autoreactivity deletion. We can manipulate cleverly to have this transgenic B cell receptor either in the presence of a certain class 1 as a self-antigen, or with that class 1 not being present by appropriate breeding. And note the analogous way, analogy to what was done in the transgenic T cell receptor to demonstrate T cell deletion. So it's kinds of clever experiments. So these mice were transgenic for the B cell receptor against H2K of K. So the idea is, if these are bred into the K background, then they should be deleted or something. So here, these are mice. They themselves are of the D haplotype, transgenic for the anti-KFK, specific BCR. So they don't recognize the self-KFD, of course. So these should be happy. They go along, they make these B cells, are making this B cell receptor, which is not recognizing a self-antigen, and therefore they mature and go to the periphery. Okay, does that make sense to everybody? So in contrast, in this, shown here in the middle figure here, now if they're bred into a background where K of K is expressed, now that becomes a self-antigen, recognized there, and in the periphery, no mature B cells are seen, or no mature B cells expressing the anti-KFK BCR. Okay, so that's recognized the self-antigen, beautiful experiment, beautiful clean experiment going against two haplotypes. So what are fates of these cells? And that so in this experiment, some of these cells die due to apoptosis, as we expect. But interestingly, some B cells were found in the periphery, and they were analyzed to understand, so we can understand how they escape this this checkpoint, and they were found to have new light chains due to this phenomenon called editing, in which there's been another recombination and another VJ event now with recognizing a different antigen, not recognizing self-KFK. So, looking at total antibody in the serum and the number of cells with a membrane-bound anti-self. Oh, sorry.
H2DD haplotype?
It should, yes. K of D and K of versus K of K.
And capital K?
So capital K, yeah, so good question. So when I say K of K, this K is a locus, and this K is a haplotype. So every mouse has a K gene of different haplotypes. Yes.
So is the fact that they're both K a coincidence?
Yes.
Okay.
Yeah.
That is very confusing.
It's meant to confuse you. Well, I think every once in a while the immunologists get together and they try to, they work on their nomenclature to make things confusing for people. I think I'm
So
firmly convinced of that. That must be a plan, they have the confusion meeting.
So if we see a big K like that, it's always referring to some random locus?
It's not a random locus, it's a predictive locus.
It is, okay, so
Oh,
K
yeah.
is a predictable locus. Could it also, is it just what the book chose, or is K always?
No, that actually was
that is
always
a
used in this
specific-
experiment.
Okay.
Yeah. The one place where they were, well, there was one experiment in the previous chapter where I think they used A and B kind of freely like that, just like, but no, this is a particular, so no, this is one of the class 1 genes in mouse.
Okay. Thank you.
Okay, but good question, on that. And yeah, and it said K of K.
Yeah.
Okay, that's the K of K. It has the K of K, the haplotype is K. So this, let me say they were D, So originally the transgenics were made on a D haplotype mouse, and then they bred them with a K haplotype mouse.
Are these used frequently in other experiment designs? Because I feel like I've come across K of K several other times.
K is used, and I'm embarrassed, I wanna say maybe C3H mice are,
Okay.
but yes,
Okay.
yeah, the K. So, I think still, one sees mostly valve C mice with a D haplotype and V6 mice with a B haplotype, but, yeah. So recall when we were talking about the synfecal peptide in the ovalbumin system, that was presented by a K of B, right? That was a K of B restricted response, so that's K again. Yeah, no, I'm glad you brought that up because I know it can be very confusing, and that's how you say it. Yeah, K of K, D of B. Okay. Yeah. And what else? Yeah, but it's not just like locus X, you know, that is a particular one. Okay. Yeah, you can ask for the Hutchings to see a K mouse. I forget. I want to say C3H might be K haplotype. I'm not sure. And of course, in humans it's different, right? Yeah. Okay. All right. So in non-transgenic expression of the anti-H2K of K transgene, no. In the transgenics that didn't have K of K as a self-antigen, so here's H2D transgenics, now there's expression and of the membrane bound and also secreted a lot, but in the transgenics that now were mated with a K, such that a K haplotype animal, such that the progeny are heterozygous for D and K haplotypes, now you do not see the expression of the transgene on the membrane or secreted. Okay. Okay. So, yeah, nomenclature is and I don't know if there were specific reasons to use this kind of, this haplotype. I mean, presumably this would have worked well, but there's sometimes things are a little cleaner with one system than another, and I don't know why. It's possible that lab used those mice for everything they did. Okay. So they looked at both presence of those cells of that antibody on the surface, as surface antibody, and also secretion, and found neither. And then in the experiment that's not shown here, where they found some escapees, if you will, when they found some B cells, they found that they had undergone editing. So that's kind of a cool answer. So this works well, this deletion, well for antigens expressed in the bone marrow. And of course, class 1 is going to be expressed everywhere. But there are other measures that turned out to be important for other antigens, including soluble antigens. And I guess I'll continue a little bit here. So completion of B cell development occurs in the spleen, again, one of the secondary lymphoid organs. So the immature Bs, that we call them immature, they leave the bone marrow, they go to the spleen under the influence of a receptor for sphingosine 1-phosphate. And these, now there are two subsets defined here, T1 and T2 for transitional between the immature and the mature conventional B2 cells. Yeah, it's called T1 and T2, but they're not T cells. So these progress in the spleen. These will become B2 cells, are sort of normal B2 cells. The normal cells we refer to as B cells. When they become mature B2 cells, they now have high levels of membrane-bound IgD. Yes.
Quick question. What does the T in T1 and T2 stand for?
Transitional.
Transitional, thank you.
Yes.
Oh, that's up top.
Not to confuse with T cells. And here's a discussion of surface markers on these these three types. So membrane IgM from early on is high. By the time they're mature, it's now intermediate. Membrane IgD is minimal, and then gets higher, and then becomes high in mature B2 cells. And some of our other markers are followed here. I guess it's showing the ones that that change during this course, during these events. The pan B cell markers, B220 and CD19, are present throughout. So these T1 and T2 groups, they form in the spleen or develop in the spleen, and they undergo selection for both survival and against self-reactivity. So these guys, T1, still being screened, as they say here, and then at the level of T1, and the possibility of negative selection is lost as they go on to T2. T2 now express a receptor for a B cell survival factor, and it says progression supported by a variety of responses. So this order of, yeah, sorry.
Sorry, for the surface marker, you said CD19 and what else?
Oh, B220, those are good common B cell markers. So the order of development has been ascertained by adoptive transfer. And again, this is one of the key things that was used in the Hardy method for the earlier development, but I think it's good to remember that that's one advantage of adoptive transfer, that one can see if one adoptively transfers one population, then one can see what they give rise to, what comes first, what has to come first, or what normally comes first. Okay. So most transition T1 to T2 occurs in the spleen, though it says in the book that some are already T2 as they exit the bone marrow. The greater maturity of T2 is associated with recirculation and entering follicles. Okay, I think I'll stop at this point, and I want to bring up another topic. So continue on these development in the spleen and this negative selection at this point. So I think I set up week 10 for a week of doing a journal club, and believe me, I have tossed this around in my mind a lot, and I think what we will do is do 3 presentations, 3 groups, each having 3 people, or 3 different papers, which I have now posted. And Pardon me, I shouldn't have done that. I want to recover that to be able to present the papers to you. So I picked 3 papers from very recent from the journal Immunity, and I chose them based on being interesting, topical, having a lot of figures that we could go over, and a little bit of variety in what we're doing. One thing I struggle with, I don't know if you've faced this in your other classes where you've done journal club, is so much of the data in immunology is big data, as they say, you know, capital B, capital D. And I didn't want to, if you're, you know, it's good to have some familiarity with the raw data before you move to the big data. And so much of immunology is that, and that's something I still struggle with, but we'll get through that.
Where did you say the papers were posted?
Oh, there's linked on the homepage. I'm gonna have that in a minute. I shouldn't have turned off the system. Oh, there we are. So, here are the links for the 3. Briefly look at what they all come with really nice, colorful graphical abstracts. Okay, heterogeneity and plasma cell lifespan. We don't care about plasma cells. Plasma cells are the B cells most advanced stage. It secretes large numbers of antibodies, responsible for long-lasting immunity. Time-stamping approach to look at turnover and how long they last. So they say that antibody secreting plasma cells have very diverse lifespans. A small fraction lasting more than 60 days. So of course, very relevant for our COVID modern times, or looking at the COVID response. Okay. So that is one B cell paper here. Talk about a particular transcriptional program. Paper number 2. Straub, recruitment of epitope-specific T cell clones with a low avidity threshold supports efficacy against mutational escape upon reinfection. Again, very relevant for our COVID response. We think a lot about these days about escape variants, especially of viruses that spin off a lot of variants. And when they do, we've seen evidence for and against initial vaccines being effective against new variants. And here they discuss low avidity to the initial challenge, initial antigen cells that are still recruited and can then be, at least theoretically, perhaps be a better fit for variants that may arise later. High avidity CD8 T cells provide protective immunity. However, their recruitment into immune responses alongside lower avidity is not well understood. High avidity are selectively expanded, but they're also, so high avidity is good, but low avidity is there and is going to be important for future responses. So that's what's discussed here. Zhao, B7. This one intrigued me. So one of the important interactions, of co-stimulation, which we haven't talked about yet, but we will soon, is between B7 on an antigen presenting cell and CD28 on T cells. Co-stimulation is a check on inappropriate activation, in that if a presenting cell has been stimulated itself, has been activated or received signals from cytokine signals, then it can fully stimulate. Here they see cis B7 CD28 interaction. In other words, both cis, in that both molecules are on the T cells, and this is due to invagination in membranes. And they believe this might explain how, help explain some anti-tumor T cell responses, and how T cells sustain their, it says, functionality in APC sparse tissues, which is a concern. So I think all of these address burning questions. B7 ligands expressed by BCs activate co-stimulatory receptor CD28. However, in peripheral tissues, APCs expressing B7 ligands are relatively scarce, raises the question of how this co-stimulation occurs. Okay, so I hope these will all be interesting to you, and the one that you present versus as well as the ones you don't present. I had to think about groups, and I decided that I am going to define the groups. Hope that's okay. And I want you to ponder for a little bit and ask and think about what paper you would like to present. And if more than one group, if there's multiple people wanting to do a certain paper, I will perform some random creation of a random number on my calculator and an award to one person, one group. So, you know, I feel like I'm at an awards ceremony, and but that's what I'll say. So I put some thought into that, and I think that will work out well. Do you know everybody's, so I put you, her name was Dendron, who's here, and Joyce, you know, Joyce, so it's Joyce there. Everybody else knows everybody else, is that correct? Tell me if you don't, yeah, okay. Alrighty, so I hope, I apologize for the kind of heavy-handed way of saying, okay, these are your groups, but I really think that anyone can find some interest in all those papers, and I certainly did, and I think that'll be a good experience. So I believe that's week 10, this is week 7. I want to present them as early as possible. And I will give you more guidance on that. I want people to be analytical, critical if necessary, but this is not either the person presenting saying this paper sucks, so or you know, none of these are going to be like that, or the other people watching saying this presenter is not good. It's more like we're all figuring this out together, and one is a leader. Okay, that's any of these papers. It's common when I'm looking at these things to have to really pay attention, and sometimes I think I understand something and I say, oh no, that's not really what they did. So don't be shocked at that at all, and that's kind of how we're going to go about it. We're going to all be going down the path trying to figure things out, okay? So we'll be asking questions like, what are they trying to show? what did they really show? It may be in the back of your mind if you can think of, well, are there other ways to approach this? Are there any experiments that they maybe could have done that they didn't do? What things cry out for to be done next? Because that's what, when you're thinking clearly and really into something, that's one of the things that comes up. What would be done next? What does this mean? What does this rule out? What does this mean for next? Okay, so I will, whenever you want to, someone member from a group can propose, you want to do a certain one, but if it's likely that if two groups ask for the same one, then I will do some random picking. Okay, questions? So we're a little distance away, but from it, yeah.
When are we like posting our choices? Like
Oh,
right
you
now,
can tell
or-
me anytime you want,
Okay.
by email or anything else. What else? So I think 3 people in a group is a good number to have, and divide as you like. There should be plenty of figures in all these. Immunity has fairly long papers with lots of figures to look at. Okay, and again, I think these are topical. At least one kind of goes against dogma, and 2 are very relevant for our COVID responses, and a 3rd for response to tumor. Okay, so I need the questions right now. You have plenty of time to go over this, but I hope it'll be a good learning experience for you guys. I think what we'll do is have one group present on one of the days and 2 groups present on the other. People always want to be later, but I think that will work out well. Okay. Any questions about anything? Of course, right now. Okay, I'll let you go. Welcome any questions.
I guess for the presentation, do you just want like a PowerPoint, or how would you?
PowerPoint is good.
Oh,
Is there another alternative that?
I mean, like, because in other classes we discuss the paper together, kind of thing. Like, so should.
Oh, I mean,
guess
I
it's
want
wondering,
there
we
to be
all
discussion. I want there to be discussion. Oh, yeah. Yeah.
I mean, so I guess everyone's gonna read.
Did you, other class, did you discuss as a group, everybody together
Yeah,
before?
like everyone
Oh.
read like one paper, and talked about kind of, I don't know.
But no, I definitely hope there would be discussion. So, yeah, it isn't all on, it shouldn't be all on the person or the group presenting, but I hope everybody will be engaged and participate. And then, yeah.
How long do you want the presentations to be?
Well, I would think most of an hour.
Okay.
Yeah, and I would like, I'll talk about this, a little bit of, what was known beforehand. And then a little bit at the end about what this leads to, as well as individual, not simply going to, well, figure 1 this and just coldly presenting that as though it's separate from everything else. I have a document that I prepared in the past with characteristic phrases that I think I like to use. So that'd be good. So remind me of that. I'll do that. Okay. I hope this will be a good experience. Yeah, this journal, Immunity, it's from Cell Press, and they are known for a glossy, not quite being exactly like a popular press magazine, but they kind of brought some of that kind of background to scientific things and having a little, nicer paper and a little brighter display, and so it became kind of a glamorous, kind of a sexy journal, you could say. Yeah. Well, journals, they compete for the best papers, right? It's a thing like that. Like Nature, if you're Nature, you want people to send to you the best paper. And Immunity, Cell-Press is a little bit a rival of Nature, but was arguably the best immunology-only journal. There's now Nature Immunology, that Nature has their own. And of course, a general, something that's really, really important could be in the general Nature or in Science as well. But again, recognize these scientists compete to get their papers in the best journals, and the best journals kind of compete, so they want the best papers to be sent to them. Okay, but I digress, okay. All right, very good. So you guys, let me know any your thoughts or your feelings. We can meet with each other now if you want to.
Yeah. Just, I don't really have a preference for the papers, so whatever you and Jake decide.
Yeah.
Okay, I'll take your hand. They seem pretty.
Want to go?
Comfortable.
Good?
Yeah.
Good, very good.